Okay, it's 10 a.m., so we'll make a start. So good morning, everybody. Thank you for taking the time to attend today's webinar on quality, ensuring the accuracy of the results. I'm Liam Feasey, and I'm the Quality and Business Services Manager here at Lextox, and I'll be running through today's webinar for you. Before we start, just a bit of housekeeping. So all attendees have been set to mute today. So this is to reduce any background noise and to ensure that everyone on today's call can actually hear me. It is a live broadcast, so unfortunately we won't be able to take any questions during the webinar. However, if you do have any questions you'd like to raise, please email them to us at experts@lextox.co.uk and we'll be happy to send a reply. And also my email address is there on the front of the slide there, so happy for you to email me directly as well. Today's webinar should last approximately 30 minutes. It is being recorded and we will share a link with you post-event as well. Okay, so let's begin. So today's webinar, as I said, is ensuring the accuracy of results. So I'll start with an introduction around how we ensure reliable results at Lextox. Then I'll go through understanding accreditation. So what accreditation is, who UKAS are, and then the various standards that we're accredited to and what testing laboratories should be accredited to. I'll then go through the quality processes that we employ at Lextox to ensure reliable and accurate results, as well as the chain of custody processes that we adhere to ensure the integrity of the sample. Okay, so ensuring reliable results. So quality underpins everything that we do here at Lextox. So we have standard operating procedures and chain of custody procedures that ensures that checks are put in place at every part of the process. So when a hair sample is received at our laboratory, one of our reporting team will match our client's instruction to the sample pack and to the sample that's received using a unique identifier that is specific to that sample and then follows that sample all the way through the process. So when the sample then enters the laboratory, one of our analysts undertakes a second check to ensure the integrity of the sample is maintained. So we continue our checks all the way through the sample as it progresses through the laboratory. So checking at various stages of the loading and sample transfer stages during the extraction of the hair sample. The sample sequences are checked on the analytical instruments by two different independent analysts, with results being processed by one analyst and then approved by another analyst. So this ensures the reliability and the integrity of the analytical result. And all of our laboratory analysts are scientifically educated, and we do work to write first-time quality assurance procedures. So accreditation. And who are UKAS? So UKAS is the United Kingdom Accreditation Service. So they are the sole national accreditation body recognized by government to assess against internationally agreed standards organizations that provide certification, testing, inspection, and calibration services. So Lextox is a testing laboratory. So UKAS attend our site and assess us against an international standard for competency. So UKAS is an independent, non-profit organization which fulfills a public role. And having UKAS accreditation demonstrates our laboratory's competence to ensure reliable and consistent analysis results. So having that accreditation provides confidence in the service and the competency of our laboratory. So Lextox is accredited to the standard 17025. So this is an international standard specific to testing laboratories. So 17025 details a number of clauses that testing laboratories should comply to ensure the reliability and the accuracy of the laboratory. Now, other um, standards exist, say for example 9001. So this is a quality management system that can be adopted and adhered to by businesses, but the 17025 is specific to technical testing and calibration laboratories. So 17025 is the accreditation mark that you should really look out for when instructing a testing laboratory such as Lextox. So Lextox has maintained 17025 accreditation for 10 consecutive years. And we are accredited for a wide range of drugs in hair. And at the time, we were the first laboratory to attain 17025 accreditation for hair alcohol testing. So the accreditation is the ultimate mark of quality and accuracy of analysis results, and it is the recognized standard for hair, drug and alcohol testing laboratories that laboratories should be accredited to. And you can see the logo that really shows and is the mark of our accreditation on the slide there, with our testing number 7516 underneath. So as well as 17025, which is the standard for testing laboratories, UKAS have also released an additional framework called Lab 51. So Lab 51 is an extension of 17025, and it really provides a more in-depth technical framework of which laboratories undertaking toxicology samples should comply to. So it really is specific to toxicology laboratories in the UK. Now, we're one of the first hair testing laboratories to actually have confirmation from UKAS of compliance to Lab 51. And it's something that we're very proud here at Lextox to have achieved and to actually conform to those quality standards. As well as the standard of UKAS 17025, we also follow the guidance of the Society of Hair Testing. So the Society of Hair Testing is an international body of hair testing experts, and all of our experts at Lextox are members of the Society of Hair Testing. And we also contribute to those guidelines and to those recommendations of the Society of Hair Testing, and help set down internationally agreed consensus. So by complying with the Society of Hair Testing consensus, Lextox is providing hair analysis within internationally agreed recommendations. Okay, so I'd like to go through some of the quality processes that we have in place at Lextox to ensure the reliability and the accuracy of our results. So the first is that we have a very robust quality management system. So a quality management system is a formalized system that documents every process, procedure, and has responsibility for achieving our quality objectives. So the QMS helps coordinate and direct our activities to meet our clients' needs and any regulatory requirements. So it really is the overall system that is in place at Lextox, that all of our procedures and all of our processes are contained within, and it's something that everybody in Lextox works within and conforms to the quality management system. And that allows us to work to write first-time quality assurance procedures. So it really is the backbone of everything that we do here at Lextox. So we have a set of operating procedures. So these are set procedures covering both technical and administrative procedures, outlining the process and the procedure of every single procedure that we do. Every sample has a unique laboratory identifier known as an LX number. So this is the number that stays with that sample and is what's used to identify that sample all the way through the process. From the moment that that sample has been collected So that's when that LX number is assigned to that sample. So then from receipt into the laboratory right through to issuing the analysis result, every sample is identified by that unique number. And we have strict chain of custodies in place to ensure the integrity of that sample. And that's traceable all the way through from the moment the sample's collected, again, right through to the issue of the results. So we have a number of quality assurance procedures that we must comply to. So there's an internal auditing of all of the standard operating procedures. So every single procedure that we have is audited internally. So this internal auditing process ensures that the procedure is correct, it ensures that it's followed, and it also can identify any improvements that need to happen with that procedure. So it really ensures that the procedure that that's in place is fit for purpose. Now, we've got a number of internal auditors that are trained by UKAS to undertake the internal auditing of our own internal standard operating procedures. We're also audited externally by UKAS. So UKAS will come on site and independently audit us against our own quality management system, against our own standard operating procedures, but also against the 17025 and Lab 51 standards to ensure that we are complying with all of the requirements of those standards, to ensure that we can then therefore maintain our accreditation and to ensure that we are issuing the correct and accurate result. We also have internal quality control monitoring. So with every batch of samples that are run, we run quality controls. So these are samples which have a known concentration of a compound, of a drug, of an alcohol marker, and the quality controls must be within a set limit in order to prove that that run has actually successfully run. And we monitor the performance of those quality controls over a period of time. So a run will only pass if it passes the quality control measures that are in place with every single run. So it's another way of ensuring that the accuracy of that run. We're also involved in external quality control monitoring, and this is done via proficiency testing schemes. So in this instance, we receive hair samples from independent organizations, and it has an unknown concentration of a drug. So we don't know what concentration of drug is in that hair sample. The independent organization will know that concentration. So we run that hair test, that hair sample, through our normal process and report the result independently to that proficiency testing scheme. They then mark us, and then we can see how we're performing. So it's a really good way of, again, checking the performance of our laboratory to make sure that we are issuing the correct result and that our processes and analytical procedures are extracting the drug and the compound as they should be. Now, with every run, and before every run, I should say, every analytical instrument, it has a system suitability check prior to that running. So this really checks the performance of the instrument before that instrument is run. So it's a very in-depth check to make sure that the instrument is running, it's tuned, and it's ready to run that batch of samples so that when those batch of samples are run, we've got confidence in that run and we have confidence in the analytical result that comes off afterwards. And there's a very strict reporting criteria that every single analytical run has to meet before it's approved. So this includes the internal quality controls that are run with that batch of samples. It includes the calibration that is used for that batch of samples. Blanks are also run. And we also look at the peak of every sample. So when a sample is run on an analytical instrument, the end result, if it's detected, is a peak. So we would get a peak showing the concentration of that drug. So we assess the peak, and we also assess the internal standard, which is run with those samples. So the internal standard is another check, and therefore, and again, we should see a nice peak with that internal standard. So there's a number of strict reporting criteria that has to be met in order to approve the analytical run, and thus give us confidence that that result is correct. And we've got strict training protocols to ensure that our personnel are competent to undertake that task. So when staff join us, they undergo a very strict training protocol, and that ensures the competency of those individuals. And every individual has to prove competency of that task on an ongoing basis. And UKAS, when they assess us, really assess the competency of our personnel, and that everything is being documented accordingly. Okay, so moving on to the chain of custody. So the collection process is an integral aspect of the whole of the sample analysis process. So right from the very moment the sample is collected, that's arguably one of the most important processes, because it's the actual collection of that sample at that moment in time, and preparing it therefore for the analysis at a later stage. So all samples should be collected under strict chain of custody procedures to ensure that the results are legally defensible and can be used in court. The majority of the time, samples are collected by our own trained sample collectors. Sometimes they can be collected by a third party, and we do provide instructions on how to collect a sample and follow those strict chain of custody procedures. We do ask that donors provide a form of identification at the time of the sample collection, so that we can verify the ID of the donor. But we also take a photograph at the sample collection, and that photograph is then included in the expert report and the results afterwards. As I said towards the beginning of this webinar, a unique identifier reference number is assigned to every single sample, and this is assigned at the moment the sample is collected at the sample collection process. And the donor must consent to the sample collection by printing and dating and signing the chain of custody form. Without the donor's consent, we can't proceed with the analysis. And if a witness is present, then they can sign and date as well to say that they've witnessed that sample collection process. Okay, so our sample collector will sign their statement on the chain of custody form to state that they've collected the samples and that they've collected them and labelled them correctly. The collection technician also states how the donor's identification has been verified: passport, driving license, for example, or any other photographic ID, as well as the photograph that is being collected. And the chain of custody form includes sample receipt integrity checks. So at the beginning of the webinar, I explained how the sample comes in, is checked by a member of our reporting team, and then goes on to second check by a member of the laboratory. So at that point, we're really looking at the integrity checks of that sample pack to ensure that there hasn't been any loss of integrity during transit of the sample from collection through to us, and also to ensure that all donor initials are present, signatures are there, and that everything is maintained before we then undertake the analysis. So then when the sample is received into the laboratory, the method of delivery is also recorded to ensure the full chain of custody is adhered to. So the whole of that process is documented, is fully traceable, and can be fully audited at a later stage. So it really does give us confidence of the integrity of that sample, right from sample collection through to issuing the analysis results. Okay, and then just to conclude, so Lextox works to write first-time quality assurance procedures. We follow strict chain of custody procedures to ensure the results are legally defensible. And we are 17025 accredited, which is the technical framework for testing laboratories. And we also have Lab 51 compliance, which is specific to toxicology laboratories. And we've been awarded 17025 accreditation, and we've maintained that accreditation for 10 consecutive years. So I hope today's webinar has really given you some insight into how Lextox maintains its integrity of the sample, how we ensure the quality of the results that we issue, and how we ensure reliable and accurate results, so that and give you confidence that the result that we issue is the correct result. So this webinar is part of our bite-sized series covering a number of testing options, information around sample collection and expert reports. And then our next webinar on the client experience, what to expect from your dedicated client manager, will be held on Wednesday, the 20th of July at 10 a.m. So I really do hope that you'll be able to attend us for that webinar, where we'll be joined by two of our experienced client managers, and they can talk you through what to expect from them. So I really hope you enjoyed today's webinar. So that concludes today's event. Please feel free to get in touch if you do have any questions. As I said, you can contact us via experts@lextox.co.uk, or you can use the email address which was on the front of the webinar, which is my personal email address, liam.feasey@lextox.co.uk. And you can also follow us on social media for any updates from Lextox, such as Twitter, LinkedIn, and our YouTube channel. Okay, thank you ever so much for your time this morning. And that concludes today.